Hey everybody, welcome to Chapter 4. In Chapter 4, we're going to talk about prokaryotic diversity. So we're going to look at all the different habitats that prokaryotes, or we're going to talk about the habitats where prokaryotes are found. And the great thing about these prokaryotic organisms is they can be found pretty much everywhere on Earth, even in environments where humans and other animals cannot survive. They are very flexible metabolically, so they can adjust to different environments for their nutrients. An example is Schwannella here. You can see her, and they're not female or male, but I just say him or her. Schwannella lives in deep ocean water where there's very little oxygen and has these long appendages that stretch out and look for oxygen. So it needs oxygen and it reaches out and it gets all the available oxygen in the environment so that it can survive. It also can shock individuals, so it has a protective mechanism about it. So they can live in environments like the Dead Sea. So here, this is the Dead Sea, and this is Halobacterium selenarum. And not only do they live in the environment, but they help to bring nutrients into that environment. So Halobacterium will decompose, will break down dead brine shrimp for their own nutrients. And the waste that they release, materials they release, actually feeds the young brine shrimp. So they're helping to maintain that environment, which otherwise would not be available to them. They're they are vital to the survival of all of our ecosystems. So they recycle elements. Just like I said about the brine shrimp are composed of elements, and when you break them down, you release the nutrients for our young brine shrimp to gain the nutrients they need to survive. So they help in recycling elements. They fix material, and fixing nutrients is just making the nutrients usable by other life forms. So like rhizobium fixes nitrogen for bacteria or for plants to use, and that's what you're seeing here. So the nitrogen in the air, nitrogen gas, they will fix to ammonia, and then plants can use this. And over here you can see this is actually the plant itself, and they have these little tiny cell structures called bacteroids, and these are kind of like structures in the plant where bacteria survive. They also degrade toxic chemicals to keep the environment healthy. So you've heard of oil spills that happen. A lot of times they clean these oil spills up using bacteria that take oil and utilize it for organic material that they can then break down for their own nutrients. So when we talk about prokaryotic organisms, they do live in communities. A community is a group of organisms that interact with each other. So we're looking at populations of different species that interact with each other. Cooperative interactions tend to be beneficial to both populations. Competitive interactions tend to cause resources to become more scarce for both populations. So basically you have two populations that are fighting for the same resources. And a lot of times that can cause both populations to be less healthy. So microbial ecology is the study of these interactions between populations and the surrounding environment. So let's look at some of the interactions that they have. Symbiosis, in a nutshell, is any interaction between different species within a community. So symbiotic relationships are just two organisms living together. Symbiotic relationships aren't always beneficial to both. They aren't always detrimental to both. So there are different types of symbiotic relationships. So there's a mutualism. Mutualistic symbiotic relationships occur when both organisms benefit. So an example would be E. coli and the human. So E. coli live in our gut and utilize the nutrients that we take in. So they're gaining benefit. But they help us by producing certain vitamins that our bodies couldn't produce without the E. coli. Amensalism occurs when one organism is harmed and the other has no effect. So an example, Prokaryote acnes lives on our skin. It doesn't really get a lot of benefit, but it can cause harm to us at certain times. Commensalism occurs when one organism benefits, but the other organism is unaffected. So Staphylococcus or epidermidis lives on our skin and doesn't really do anything for us, but gains benefit from the nutrients on our skin. Now, I would argue that Staph epidermidis and humans actually have a more mutualistic relationship because they are one of the most common bacteria on our skin. And so their being there on our skin causes other organisms to not be able to settle. So pathogens can't get a foothold into our integument because of them. So in that sense, they are mutualistic. Neutralism occurs when neither gain benefit. So endospores have a neutralism relationship. They don't cause harm, they don't benefit. They're just kind of there. But when the environment changes, then they can germinate and then they could potentially cause problems. And parasitism occurs when one benefits and one is harmed. So these are your disease pathogens that we typically talk about, and again, humans would be the ones that are being harmed. The pathogen is gaining benefits. So this figure actually comes from a textbook from Anatomy and Physiology, in McKinley textbook that we have used in the past. And it shows the microbiome of humans. So humans have both resident microbes and transient microbes. Resident microbes are always present, whereas transient microbes come and go depending on the age of the organism a lot of times, or the health of the organism. So an example of transient microbes would be microbes that come during puberty. Prokaryote acnes is more likely to be found in your skin during puberty and can cause serious acne problems in teenagers. Whereas in adults, you're not likely to find them. But Staphylococcus aureus is continuously found on our skin, in our nasal cavity. Streptococcus is found in our mouth, our oral cavity, in our nasal cavity. These are always there. So those are resident microbes. Transient microbes come and go. So human microbiome begins at birth. The womb is actually a sterile environment, so the only types of cells in the womb are eukaryotic human cells. But as soon as that baby is born, moves through that vaginal birth, the baby gets covered with bacteria. And as the baby drinks milk from mom, or gains nutrients in other means, plays with toys, touches stuff, puts his or her mouth or hands in their mouth, they gain bacteria. And each time they gain bacteria, they are just adding to their own microbiome. So here you have the visible human, but if you were to take away all the visible stuff, and you would see that we have millions upon millions of microbes. We have approximately 10 times more microbial cells than we have human cells. And so that's that complete human there. And these microbial cells are really important. They actually help us to produce all the nutrients we need. They help to protect us, and they help to produce our immune system. So they're really important. So now I'm going to stop here. We're going to get in our next video, we're going to start talking about classifying organisms into different groups, prokaryotic organisms into different groups. The way we classify organisms in the past differs from how we can classify now because we have more tools in our classification arsenal. But we'll talk about that in our next video, okay? Bye.